Hello everyone. I'll introduce the paper submitted to IMETA. The title is Cross-Tissue Multi-omics Analyses Reveal the Gut Microbiota's Absence Impacts Organ Morphology, Immune Homeostasis, Bile Acid and Lipid Metabolism. Germ-free mice display distinct anatomical and physiological traits, which underscore the essential role of the microbiota in host development and homeostasis. Our study highlights 4 key findings. 1. Construction of single-cell, spatial transcriptomics, and bile acidomics atlases in germ-free mice. 2. Pronounced aberrations and tissue-specific heterogeneity in B cells, myeloid cells, and T/NK cells. 3. Microbiota-mediated shaping of mucosal zonation and modulation of lipid dynamics in the small intestine. 4. Anomalies in liver bile acid synthesis and ileal reabsorption in germ-free mice. Single-cell RNA sequencing was conducted on 8 tissues. After quality control and filtering, 269,105 cells were retained for unsupervised clustering analysis, with 85.2% identified as immune and epithelial cells. Spatial transcriptomics revealed tissue-specific aggregation behaviors of plasma cells, microbiota-mediated enrichment of Kupffer cells around the portal vein of the liver, and a reduced aggregation index of the splenic marginal zone, marked by the Marco gene. Our study revealed key abnormalities in B cells of GF mice, including aberrant proliferation, differentiation, and activation, particularly in splenic marginal zone B cells. Further analysis showed that the fate of splenic follicular and marginal zone B cells is regulated by complement receptor 2. Our study demonstrates that the microbiota regulates neutrophil survival in the bone marrow, disrupts the thymic T cell progenitor development, and reveals heterogeneity in intestinal lymphocytes, with lipid absorption pathways enriched in small intestinal gamma-delta T cells in GF mice. Integrated single-cell and spatial omics analyses reveal that the microbiota influences intestinal epithelial function by regulating its compartmentalization. Microbiota deficiency increases the proportion of functional cells in the intestinal epithelial top layer and significantly thickens the epithelium. The microbiota regulates epithelial function in GF mice, characterized by decreased intestinal epithelial immune function, enriched nutrient absorption pathways in the ileum, downregulated energy metabolism in the large intestine, and enhanced metabolic function of the Reg3G high epithelial subtype. As far as intestinal epithelial immunity is concerned, it appeared that GF mice have adaptive immune deficiencies associated with antigen processing and presentation, while innate immune activation associated with antiviral activity was maintained in GF mice. Our study reveals unique lipid droplet characteristics and autophagy features in GF mice. Additionally, we demonstrate for the first time that the microbiota regulates the expression of LPL-ZBTB20 and its zonation within liver lobules, contributing to lipid metabolism regulation in GF mice. GF mice exhibit reduced bile acid concentrations in multiple tissues and significant changes in hepatic bile acid composition, suggesting a shift in bile acid synthesis pathways from the classical to the alternative route. Key genes involved in bile acid synthesis and enterohepatic circulation reabsorption are also significantly altered in GF mice. Our study provides a multi-organ, multi-omics atlas of SPF and GF mice, revealing microbiota-dependent plasma cell aggregation, impaired B cell maturation in GF mice, microbiota regulation of T cell development, altered gut mucosal zonation with enhanced lipophagy, and microbiota-modulated liver lipid and bile acid metabolism.